Hello, all. In this video, we'll be seeing about nucleic acids. Nucleic acids are essential biomolecules that store and transmit genetic information. These nucleic acids are made up of amino acids, and the nucleic acids are categorized into two basic types: DNA and RNA, deoxyribonucleic acid and ribonucleic acid. The DNA is helpful in storing the genetic information, and RNA helps in the synthesis of proteins. So let's see a few applications of these nucleic acids. So DNA vaccine for rabies. DNA vaccine is a type of vaccine where we extract the DNA of the bacterial or the viral pathogen or the agent and then induce it into the cells. And these DNA will react with the cells and help in producing the bacterial or the viral proteins which come to the surface of the cell. And once these proteins are outside on the cell surfaces, the immune system would activate a certain type of antibodies which will help in fighting against these proteins and increase our immunity. So let's see how the DNA vaccine for rabies works. The DNA vaccine for rabies, we extract the DNA from the rabies virus and then we induce it into the cells. And once it is introduced into the cells will start reacting to it and then they produce the bacterial proteins for the rabies DNA. Once these proteins are settled onto the surface, the immune system will generate a type of antibodies which will act to these specific proteins and then they start increasing antibodies uh, in our body. The main use of these DNA vaccines for rabies is because they are advantageous to be stored and they can be injected in any way. Either you can give it as an injection or orally also, and then they also have a long-lasting effect. So these are the advantages for using a DNA vaccine in rabies. And let's go further. So now RNA vaccines for COVID-19. So in RNA vaccines for COVID-19, we extract the RNA of the virus and then we induce it into the human body or the cells. Just like the DNA vaccine, where we take up the DNA of the bacterial or the viral pathogen and then inducing it into the cells. Over here, we'll be using the RNA. And these RNA vaccines are actually effective in COVID-19 because the RNA of the COVID-19 virus, that is the SARS-CoV-2 virus, from which we extract the RNA and synthesize the vaccine for the disease. So here, the vaccine was first produced by Pfizer BioNTech company, and then it was Moderna vaccines. These two vaccines were the first one that had come up, and they were actually seen to be very effective. The reasons for using RNA vaccine is because they are highly manufacturable. That is, we can produce RNA vaccines in a vast amount in a short period of time. And this is a main criteria for times like pandemics, like COVID-19 pandemic, where we need a huge amount of vaccine supplies in a short period of time. So RNA vaccine helps in production of a huge amount of vaccines in a short period of time, and then they are also easily mutable or modifiable. That is, if there is any new variant of these viruses which are not affected by the previous vaccine, then these vaccines can be modified easily, which can be used further for the new variants of the virus. So these are the advantages of using RNA vaccines for COVID-19, and these are the RNA vaccines prepared. Let's go further. Forensic DNA fingerprinting. Forensic DNA fingerprinting is a technique in which we identify individuals based on their unique DNA profiles. It involves analyzing specific regions of an individual's DNA to create a genetic fingerprint. DNA fingerprinting is widely used in criminal investigations, paternity testing, and identifying human remains. So DNA fingerprinting, we mainly use it in criminal cases like forensics and all those things where we need to find out about a certain victim or any paternity issues where you need to find the parent of a child and all those issues. Over here in DNA fingerprinting, the first mainly what we do is we take up the DNA sample and then we try to extract the DNA from it by using a certain processes. So there are a few steps in the DNA fingerprinting technique. That is, we take the DNA sample and then extract the DNA, and then we have to multiply the DNA amount or clone it so that we have a lot of DNA samples which we can be using it for the testing. Because we cannot do the DNA sampling with DNA fingerprinting test with just a small amount of DNA. We need a large amount of DNA samples for that. So we just use certain cloning methods to increase the amount of DNA. And then by using gel electrophoresis method, we try to figure out the DNA pattern of it using a certain amount of gel called as agarose gel in this method, where we have a electric plate or electric conductor upon which we place the sample of DNA and the agarose gel, and when we apply this current to the conductor, the DNA all the antigens on the DNA will get separated with respect to their size and their antigens due to the charges that are produced by the current. And through this method, we can separate the DNA and see the patterns produced by the DNA. And then by verifying it visually, we can say whether the DNA belongs to a certain person or not. So this is how the DNA fingerprinting process works. Applications of DNA fingerprinting. They are used in forensic science, personal identification, determining paternity and maternity, breeding programs, diagnosis of hereditary disorders, the creation of treatments for inherited diseases, detection of AIDS, etc. So let's see them one by one. Forensic science. Biological samples such as blood, hair, saliva, sperm, and body tissue cells are used for DNA profiling. It is possible to compare the DNA recovered from the evidence sample using the VNTR, variable number of tandem repeats, prototype. It aids in the investigation of crimes like rape and murder. Personal identification. It makes use of the idea that DNA fingerprinting can be used to identify people acting as a kind of genetic barcode. Determining paternity and maternity issues. A person accepts their VNTRs from their parents. Cases that were in disagreement have been resolved via parent-child VNTR prototype analysis. So while in this paternity issues, we have seen that the child or the infant actually inherits the DNA from both the father and the mother. So the DNA of the child will actually be the combination of both father and mother. And comparing the child's DNA with the father and mother, we can find out the whom the child belongs to, and then solve the issues regarding to the problem. And then breeding programs, breeders typically assess a plant or animal genotype using its phenotype. After DNA extraction, restriction enzymes are added which work like scissors to cut the DNA into smaller segments that are different between individuals. Since homozygous or heterozygous dominance is difficult to distinguish from each other because of from their appearances, the genotype can be determined with great care and accuracy using DNA fingerprinting. Hunting dogs and racehorses can both benefit from it. Diagnosis of hereditary disorders. It can be used to identify inherited diseases in both newborn and parental children. Cystic fibrosis, hemophilia, Huntington diseases, familial Alzheimer's, sickle cell anemia, thalassemia, and etc. Many other diseases can be diagnosed using these DNA fingerprinting methods by finding out the specific DNA or the specific damaged part of the DNA and then changing it. The creation of treatments for inherited diseases. DNA prototypes linked to the disease can be identified by examining the DNA fingerprints of family members who have a history of a particular disorder. Detection of AIDS. A person with AIDS can be diagnosed by comparing the HIV RNA band with the bands found by the man's blood. So these are the applications of DNA fingerprinting. Now let's see the advancements done in fingerprinting technology. So advances in DNA sequencing technology have improved the speed and accuracy of DNA fingerprinting. Next generation sequencing techniques allow for the analysis of multiple loci simultaneously, enhancing the discriminatory power of DNA profiles. Emerging technology like rapid DNA analysis promise to further streamline the DNA fingerprinting processes. So these are advancements done in DNA fingerprinting. And now let's go to the conclusion of the topic. So now in this video, we saw about DNA vaccines, RNA vaccines, and then DNA fingerprinting, and all these come under the few applications of nucleic acids. And we saw the advantages of using DNA vaccines. That is, they can be used as a they have a long lasting effect, and they are not that much effective. I mean, they don't have the lot of side effects because we don't use the live virus itself. We are only using the DNA or the RNA in case of RNA vaccines. So they are uh, lesser side effects and more effective in their respective cases. That is DNA for rabies virus, DNA vaccine for rabies virus, and then RNA vaccine for COVID-19 virus. And forensic DNA fingerprinting is helpful in finding out the certain issues that we saw before, that is about the uh, solving issues between the paternity and maternity, and then finding out few criminals or victims of a certain case, that is criminal cases and all those things. So this is all for this video. Thank you.